Before we start with our lesson, I highly recommend that you prepare a piece of paper and pen to jot down important notes. Also, you may pause the video to catch up on the lesson when you need to, and you may also rewind the video. We are also going to conduct an assessment for this lesson through Kipper, so let's start. Today's lesson is about molecular radiobiology. Radiation interaction. Radiation interaction with a tissue will interact specifically at the electron level, while radiation that interacts and will result to an observable radiation injury or damage specifically interacts at the molecular level. Appearance of lesions is a result of radiation on macromolecules and on water. But what are lesions? Lesion is a region in an organ or tissue which has suffered damage through injury or disease. It says here that water is a principal molecular radiation interaction in the body. So right now in our discussion, we are going to discuss the irradiation of macromolecules and radiolysis of water. So why water? Since our body composes of 80% of water, then water is a principal molecular radiation interaction in our body. Irradiation of macromolecules. So we have several types of macromolecules. For example, we have lipids, we have proteins, nucleic acids, carbohydrates, and of course we have water, which is the most abundant one. There are two types of irradiation that can occur. We have in vitro and we also have in vivo. In vitro occurs outside the body or outside the cell, while in vivo occurs within the living body or within the living cell. Irradiation of macromolecules. So there are three major effects that occurs when macromolecules are irradiated in a solution in vitro. So when we say solution, it is a liquid that contains dissolved substances. So there are three major effects. We have the main chain scission, the cross-linking, and the point lesion. So here is a diagram that represents the three major effects. A is for main chain scission, B is for cross-linking, and C is for point lesion. So let's start with the first effect, the main chain scission. It is defined to be the breakage of the backbone of the long chain macromolecule. It results in the reduction of a long single molecule into many smaller molecules, and we have two effects. The first one is that it reduces the size of the macromolecules and it reduces the viscosity of the solution. An important note is that the measurements of viscosity determines the degree of the main chain scission. The second effect is said to be cross-linking. It is the process of side spurs created by irradiation and attached to a neighboring macromolecule or to another segment of the same molecule. So from the word cross-link, it means that it will attach to another molecule. So one effect is that it increases the viscosity of the macromolecular solution. The last effect is the point lesion. It is any change that results in the impairment or loss of function at the point of a single chemical bond, which means that a disruption of single chemical bond can lead to point lesion. It is also said here that point lesions can result in the stochastic radiation effects observed at the whole body level, which means that the cellular radiation damage that results in the late radiation of effects. Synthesis of macromolecule. Synthesis means it is the creation of something through metabolism, which can be either catabolism or anabolism. So when we say catabolism, it is a breaking down of molecular nutrients to be diffused into the cell membrane, while anabolism is a production of large molecules from smaller particles. Radiation damage to any of these macromolecules, for example the DNA, the proteins, the carbohydrates, may result in cell death or late stochastic effects. So speaking of DNA, let's talk about its synthesis. So we have three components, the deoxyribose, the phosphate, and the nitrogenous base. Now all of these three are collectively called the nucleotide. The DNA is said to be the most radiosensitive molecule due to the presence of the nucleic acids. Now, the nucleotide will now gather towards the nucleus of the cell during G1, and the DNA is in now a double helix form. So like this. Proceeding to the S phase, the DNA now will start to split up and open in the middle like a zipper. During this phase, DNA consists of only a single chain with no base pairing, but won't last long since sugar phosphate molecule attaches to the DNA single strand sequence as determined by permitted base pairing. So from one double helix, it will now result into two similar molecules which will exist. Each is a duplicate of the original, meaning magkapareho. Hence the two daughter DNA molecules. So here are the 5 responses of DNA to radiation. Now, the first 4 radiation responses will result to structural change. And the last response, which is the change in or loss of a base, may result to an irreversible damage. So here is a diagram which illustrates the types of damages that can occur in DNA. A here represents one side rail severed, B represents both side rails severed, C is for cross-linking, and D is for wrong breakage. So now we're done with the first part, which is the irradiation of macromolecules. Now let's proceed to the second part of our lesson, which is the radiolysis of water. So as we all know, water is the most abundant molecule in our body, which comprises 80%, in which it is the principal radiation interaction that can happen in our body. So a water molecule is written as H2O, and when an atom of H2O or water is irradiated, it is ionized and dissociates into two ions, which we call as the ion pair. A lot of interactions can possibly happen. One example is when the ion pair will join a stable water molecule, no damage will occur. Another interaction which can possibly happen is when the free electron may attract to another water molecule to produce a third type of ion. The two interactions may result to these. Both are unstable and may dissociate into smaller molecules. The final result of the radiolysis of water is the formation of an ion pair, which is H plus and OH negative, and two free radicals of H and OH. But what is a free radical? A free radical is an uncharged molecule that contains a single unpaired electron in the outer shell. They can contain excess energy that can be transferred to other molecules to disrupt bonds and produce point lesions at some distance from the initial ionizing event. Like for example, the free radical OH, the one with the asterisk symbol, it can bond with another free radical OH. So OH, free radical, and another free radical OH will result to hydrogen peroxide, or H2O2, which can be poisonous to the cell. The free radical H can interact with an oxygen molecule O2 to form hydroxyl peroxide, or HO asterisk 2. This can also damage the cell. Also, hydrogen peroxide can also be formed when two hydroperoxyl radical interacts. And so we have come to the last part, the direct and the indirect effects of radiation. When we say direct, it is the initial ionizing event that occurs on the target molecule, and the indirect effect is when the initial ionizing event occurs on a distant non-critical molecule and the energy is transferred to the target molecule, meaning it has to go through a medium. The principal effect of radiation on humans is indirect. Why? Actually, it is hard to determine, but given the fact that the human body consists of approximately 80% of water and less than 1% DNA, it is concluded that essentially all of the effects of irradiation in vivo result from indirect effect because it has to pass through water before it can actually damage the DNA. So that's the end of today's lesson. I hope you learned something. Please refer to your book for additional information.